Hello, everyone, and welcome to my presentation. My name is Shao de Bittenair, and today I would like to talk about the characterization of sustainable polyesters. First, a big picture overview of sustainability. As we develop and consume more in society, we need to find ways to maintain our existing natural resources in order to preserve a balanced global ecosystem. Regarding material resources, this can be accomplished through the reduction of material waste in product design and packaging choices, faster degradation of waste, recycling or reuse of already processed material, and renewable feedstocks to synthesize the polymers we use just in general. All these aspects are particularly important for polymers since many products contain them, particularly in single use and packaging. The alternative life cycle paths I have mentioned are shown in the figure to the right. For this project, we focus on the synthesis and properties of polyesters. Polyesters are simply a class of polymers which contain an ester group in the repeat unit and are commonly used as fibers in clothing and food packaging. Polyethylene terephthalate, known as PET, is a common recyclable polyester used in packaging and is known by the plastic type number 1. The repeat unit is shown to the right and the ester group is highlighted in the box. Polybutylene succinate, otherwise known as PBS, has become a promising biopolymer in recent years. It is semi-crystalline, thermoplastic, and synthesized through a polycondensation reaction between 1,4-butanediol and succinic acid, as shown in the synthesis scheme above. PBS has such a great potential because both its reactants can be produced via a sugar fermentation reaction instead of using the traditional petroleum-based feedstock. Its mechanical properties are also comparable to commercially used polymers such as high-density polyethylene and polypropylene. However, PBS is considerably more degradable than those. Our research involves synthesizing a series of polyesters similar in structure to PBS, but with various lengths of hydrocarbon chains between the carboxylic acids, as shown in the box. Therefore, we can observe trends as the polymer chains become more like polyethylene, whose structure contains entirely hydrocarbons and has notoriously poor degradation. Essentially, we are changing the acid with which to synthesize our polyester with. Polymers are often characterized by their thermal and molecular weight properties. However, surface wettability is another important property to study, particularly for biological applications. Wettability depends mostly on chemical composition at the surface and surface roughness. These factors play a role in determining the efficacy of, say, cell adhesion, protein absorption, and biocompatibility, which are all extremely important for medical applications such as tissue scaffolds. Wettability can also affect surface aging, lifespan of coatings, and overall degradation of the material. One way to change the chemical composition of the surface is using a vacuum ultraviolet photooxidation plasma treatment. Here we have a simplified diagram of the process. High energy UV photons are generated from a light source at wavelengths which cause argon gas introduced into the sample chamber to become energized into a plasma state of matter. These high energy argon atoms then collide with the surface of the material to form radical carbons. Oxygen gas is then introduced into the sample chamber, reacting with the radical carbons and bonding to the surface as a result. As the oxygen content increases on the surface, the hydrophilicity will also increase. Wettability and hydrophilicity are commonly measured using contact angle goniometry. Here we have a diagram of a contact angle goniometer. In general, the way it works is an automatic dispenser is used to place a drop of water onto the surface of the material. We use a hot press to create polyester films, which are then placed on the sample stage. A live feed camera is trained on the drop and the sample stage and adjusted such the camera can take a clear image of the drop. The software then calculates the angle at which the drop rests on the surface of the polymer film. As you can see in the figure to the right, a high contact angle above 90 degrees indicates a hydrophobic surface, and a low contact angle below 90 degrees indicates a hydrophilic surface. Contact angle is also related to adhesion, wettability, and surface free energy. The other factor determining contact angle is surface roughness, which can be measured using a non-destructive technique called atomic force microscopy, or AFM. The general setup involves a probe consisting of a nanoscale sharp silicon nitride tip attached to a flexible cantilever, which is scanned in a raster pattern. This is simply a continuous rectangular zigzag. A piezoelectric ceramic scanner controls the position of the probe relative to the sample surface. As the tip moves over the surface, the probe will then be deflected according to the surface topography, which is tracked by a laser and a position-sensitive photodetector, as shown in Figure A. This information is used to construct a high-resolution 3D image of the surface topography. The roughness of the surface can then be calculated based on the image generated. Here in Figure B, I have shown where Figure A would be happening in the AFM. So this is the sample stage and the probe mount. In Figure C, I have also shown with a reference to scale of my fingertip, the AFM tip and the cantilever. The AFM has static or contact mode and dynamic modes, including tapping and non-contact. Contact mode involves maintaining direct contact of the probe and the surface, often requiring soft and flexible cantilevers to avoid probe damage by repulsive forces between the tip and the surface. Tapping mode involves oscillation of the probe using a piezoelectric actuator such that the tip lightly touches the surface with the frequency of the oscillation and experiences repulsive forces. Thus, the probes can be stiffer and suffer less wear and tear overall. Non-contact mode also involves an oscillating probe, but at a lower amplitude such that the probe does not touch the surface and experiences attractive forces. This mode requires the least amount of sample interaction with the probe and is best for liquid samples. However, it requires precise control of the tip oscillation, therefore it becomes more expensive. Here are some example AFM images taken from the hot pressed films for PBS and a PBS-like polyester using dodecane dioic acid. You can view the 2D images in figures A and C, respectively, and their corresponding 3D images in figures B and D. Notice the color grading of the image indicates the relative height of the surface topography. Also note that the PBS-like polyester is in general appears rougher than the PBS film. Scanning electron microscopy, or SEM, is another method to image the surface of a sample. First, in vacuum space, electrons are generated at the top of the SEM column, and then accelerated and focused into controlled beam by condenser lenses, as shown in Figure 1. Scan coils then deflect the electron beam such that it is scanned over the sample surface in a raster pattern to create the image. As electrons interact with the sample surface, secondary electrons, backscattered electrons, and X-rays are produced and detected by specialized detectors in the sample chamber, as shown in Figure 2 and Figure 3. Figure 2 shows the different emissions based on the penetration of the electron beam, and Figure 3 is an overview of the entire instrument and its components. Here are some example SEM images taken of the same polyester films. The SEM images also show the PBS-like polyester has a rougher surface than the PBS film. Here's a comparison of the two instruments. The major advantage of AFM is that its magnification is in 3D, while SEM only has magnification in 2D. AFM also operates in vacuum, air, or liquid, whereas SEM operates really only best in vacuum. AFM has no sample preparation required, while as SEM samples must be conductive, ergo they may require sputter coating before measurement. Sputter coating is simply a process by which a very thin atomic layer of metal is applied to the surface of the sample to make it conductive. Some cons for the AFM is that it has a small depth of field in the nanometer scale. SEM has a much larger depth of field for rougher surfaces in the micron scale. AFM can also be expensive to operate as the tips need to be frequently replaced and they can be fairly costly. SEM can have fairly inexpensive operation as you only need to maintain the vacuum pump. AFM can measure physical surface properties, including adhesion, conductivity, and more with particular tips. And SEM can measure surface chemical composition using characteristic X-rays and backscattered electrons. Both are able to measure different properties, which leaves the determination of which to use dependent on the specific application. Here are my references for more information on the topics I discussed. And I would like to thank my research advisor, Dr. Matt Meary, my fellow research students, Jules Samonte, Amina Endelia, and Liam Riley. I would also like to thank the School of Chemistry and Material Science at RIT and the Summer Undergraduate Research Fellowship, which I received funding from. Thank you, and I hope you enjoyed my presentation.